Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I am Mr. Ulrich. And in the past couple notes cast, we've been talking about transcription and translation, that is going from the base pair sequence of DNA to a amino acid sequence of functional protein, and how we kind of turn that on and turn that off in order to go through differentiation and all that kind of good stuff. In this notes cast, we're going to be talking about how errors can take place during replication or during meiosis, mitosis, during chromosome separation. If we're going to be talking about mistakes that are made in the language of DNA, then we should probably talk a little bit about the language of DNA. Remember, it's A's, T's, G's, and C's. Those are the kind of the four letters of the language. And the words are made up of three letters, three of those bases together, forming a codon. And a lot of times we'll kind of use an analogy, use sentences in English that are made up just of three-letter words, like this one right here. And of course, you can see the pattern. I don't need to split it up, but it is a sentence about an old man in the sun and not getting his hat. So let's mess with the language a little bit. We're going to switch out one of the letters, and I made it real obvious because I made the letter red. But you can still see the sentence. It basically means the same thing, even if I split it apart. You can see that it's the sun was hot, but the old man did not get his hat. So it basically not that big of a change. This is an example of a point mutation where we're just changing a single base and replacing it with another base. A lot of times these are called silent mutations because there is no amino acid change. As we can see up in here in this example, the code for GGC for glycine is converted, mutated to GGU instead of the GGC. But because there is this redundancy in the codes, GGC, GGU, they both code for glycine, and so there is no amino acid change. Sometimes, though, we are going to see a change, like we see down here in the next example, and it changes just one base. Excuse me, that one base changes just one amino acid. The most of the protein is going to be the same. We call this a missense. Although sometimes, as in the last example, if we're going to change a codon from a codon to for an amino acid to a stop codon, then we're going to get a completely wrong protein. None of it is going to make sense, and it's going to be utter and complete nonsense. Sickle cell anemia is a perfect example of a genetic disease that's caused by a single point substitution. In this case, in the beta chain of hemoglobin, right at the sixth codon, the second base of the sixth codon is switched. And so instead of coding for glutamate, which is hydrophilic, it codes for valine, which is hydrophobic. And this changes the way that the beta chains interact with each other, changes the way that the oxygen is carried by the hemoglobin, all kinds of issues. Now, this is a problem. But we see it not very common, but we see it more common in African Americans and in African communities. What it turns out is that there is actually a selective advantage to being at least heterozygous for sickle cell anemia. In that case, you'd still make some functional hemoglobin B, and so you'd have some functional red blood cells. In the case of homozygous sickle cell anemia, you don't make functional hemoglobin B, and the cells are targeted for death. They break down because they're abnormal, and so you get a buildup of bilirubin, and the abnormal cells lodge in the capillaries and causes all kinds of problems. But again, being heterozygous seems to give some benefit to resisting malaria. It's, the malaria protozoan needs a high oxygen level in the blood in order for it to thrive. And people with at least heterozygous sickle cell anemia have lower blood oxygen content level, and so they don't carry the malaria as well. It's not always a disease state. That's the result of a mutation. In this map, we're actually looking at the global distribution in one population compared to another population of a single nucleotide polymorphism, an SNP. This one in particular, RS129138832, results in the characteristic of blue-eyedness. There's old Blue Eyes himself. Now, I'm not sure why the map makers did this, but the blue-eyed allele is actually orange, and the brown-eyed allele is the blue part of each one of those pies. So you can see up in like Northern Europe and Finland and in those regions, blue-eyedness is more than half of the population. But in other parts of the world, it is a minority of the population. Blue-eyedness is kind of cool the way that it works. Most commonly, the mutation, this particular single nucleotide polymorphism here, is actually it's a good example of gene control. Now, the mutation itself is actually in the gene for HERC2, which is a part of a protein, part of a polypeptide that is part of ubiquitin, a protein degradation enzyme. But within one of the introns of the HERC2 gene is actually the control locus for the OCA2 gene itself. Now, the OCA2 protein involves the development of melanocytes within the iris, the stroma of the iris of your eyes. And the more melanocytes you have there, the browner the eyes are. In this case, when this protein HLTF associates with the locus of control region on the HERC2 intron, it allows the OCA2 region to become euchromatin and allows those nucleosomes to loosen up, and it allows the polymerase 2 to get in there and transcribe and translate and all that good stuff. When there's this mutation in this intron, then the HLTF cannot bond to that control region in that intron, and without the HLTF bonding, the OCA2 region stays as heterochromatin and doesn't unwind, and it can't be read, and so the gene is essentially shut off. Where substitutions tend to only affect one codon, if at all, frameshift mutations affect each and every codon downstream from the mutation itself. These involve either insertions, where we're adding a base that isn't supposed to be there, or taking one out that is supposed to be there through a deletion. To show how this works, let's go back to our old man in the sun without his hat. And let's really mess with the message. I'll do a little deletion. And of course, with your fabulous brain, you're going to look across and you're going to see the sun was hot, but the old man did not get his hat. But if you were a stupid ribosome and you put on your ribosome glasses, you're just going to see 3 letters, 3 letters, 3 letters, 3 letters. And of course, this makes not a whole lot of sense. And that's why we call this a nonsense mutation. Cystic fibrosis is a good example of a disease that results from a frameshift mutation, where sickle cell anemia is a disease seen more often in African populations. Cystic fibrosis is more often in European populations. The gene involved codes for a protein, it's a transport protein that moves chloride ions across the cell membrane, and when it's defective, the mucus doesn't move as well. It's stickier and thicker and traps more bacteria, and because it doesn't move, you end up with more bacterial infections, to the point where untreated life expectancy is pretty low, and even treated life expectancy is also lower, though it keeps getting better and better and better. Without the transport of chloride, we don't get the transport of water into the mucus that's forming on the outside of the cells. And without this, the water, the mucus becomes thicker and doesn't move the way that it's supposed to, trapping all kinds of bacteria and, like we said, building up bacterial infections. The most common mutation involved in cystic fibrosis is called the delta F508, and it is a deletion of three bases. And so it is sort of a frame shift in that it spans two codons, but it does only affect one amino acid. We've been talking about changes in the base sequence that affect a single gene or even just a single codon, if at all. There are other mutations that affect whole chromosomes or whole chunks of chromosomes involving suites of genes. Quite often, these chromosomal mutations take place during meiosis through the process of non-disjunction, where tetrads that normally segregate evenly into two chromatids and two chromatids instead separate unevenly into three and one chromatid. The result of this, of course, is some of the gametes will have an extra chromosome and some of the gametes will be missing a chromosome. If those are the gametes that go ahead and fertilize the egg, then the zygote's going to have the wrong number of chromosomes. Chromosomal mutations can be diagnosed in unborn fetuses through the process of karyotyping. This is where cells are collected to see some that are going through mitosis. They're stained, and you take a picture of them and look at the chromosomes, arrange the chromosomes by banding pattern and size and shape, count them up. You can get a definitive diagnosis on not just chromosomal mutations, but certainly on the sex of the baby. Classically, the cells are collected through amniocentesis. This is where a needle is used to withdraw a little bit of amniotic fluids from surrounding the fetus, and that's where we find the cells to go through karyotyping. Now, of course, when we look at this karyotype here, some questions I hope are coming up. How many chromosomes are we supposed to have? I thought we're supposed to have 46. This has got a few extra here because this is a cow. Actually, this would be a bull. All right, no more cattle. Here we go. Here's a human karyotype. Now, how many are we supposed to have again? Because if you look carefully here, and count across, I got pairs, I got pairs, I got pairs. Wait a minute, down here at 21, I don't got a pair. I have an extra 21st chromosome. Having that extra 21st chromosome leads to a condition known as Down syndrome. It is also called trisomy 21, to talk about that triplet at the 21st position. It was first described as a distinct and separate mental disability by John Langdon Down back in the late 19th century. People with Down syndrome tend to have a set of physical characteristics as well as mild to moderate mental retardation and an increased risk of some cancers. Interestingly, Down syndrome can also be brought on translocationally. That means part of that 21st chromosome is actually attached to a different chromosome. In this case, it most often sticks onto the end of the 14th chromosome. So you still have an extra, even though it's attached to a different chromosome. The most common chromosomal mutation that is survivable involves the sex chromosome. Girls who lack a second X chromosome have what's called Turner syndrome. And without that second chromosome, they can't go through meiosis, they can't make functional gametes, and so they're sterile. They do have a few other physical abnormalities, neck webbing and clubbing of the hands and feet, but it is survivable. Whether the disease results from a gene mutation or a chromosomal mutation, we're going to want to fix the mutation. We're going to want to address the problem. The problem, of course, is that if the mutation came from your parents, if you inherited from your parents, it's in every one of your cells. And that's going to be a little hard to fix because that's a lot of cells. If we're going to keep it from being passed on to the next generation, then we at least have to repair it in the gonads. But again, we have this problem. We can't really fix the cells. If there is a gene mutation and we're missing a protein, maybe we can just address it by supplementing the protein that's missing, if that's even possible. Other than that, it can't be fixed yet. there are some ideas. What if we could engineer a groovy virus that instead of carrying viral DNA so that the infected cells just make more viruses, we could engineer that virus to carry like a payload of the genes that are actually missing. And so we could infect the cells with the genes that they're missing. It sounds like it could work, and there's certainly a lot of research going into it. We'll see what happens in the months, years to come. If these mutations take place in genes or regions of genes that are involved in mitotic control, well, now these mutations can lead to cancer. And since we can't fix mutations, we really have to avoid mutagens. Number one mutagen that we're exposed to, of course, is ultraviolet radiation. Please wear sunscreen so this doesn't happen to you. If Darwin had a clue as to what genes were or how they worked, what do you think he would say about mutations? Well, that's enough about mutations for now. Thanks again for checking into Mr. Ulrich's Land of Biology. I, again, am Mr. Ulrich, and if you have any questions, please feel free send me an email, put them in the comment section, whatever you got to do. Other than that, we'll see you in class.